Hello and welcome to the fourth day of the workshop, Mycobacterium tuberculosis complex, next generation sequencing made easy, data analysis step by step. My name is Arash Khodousi, and in the next couple of hours, we will discuss about the use of web tools and software which are available for Mycobacterium tuberculosis complex sequence data analysis. Well, here are the outline of my today presentation. In the first part of this presentation, we will review the currently available web tools and pipelines for the fast identification of bacterial strains from the raw sequencing reads. And in the second part, I will introduce to you the web tools and software developed specifically for analysis of Mycobacterium tuberculosis sequence data. Despite being a well-established research method, the use of next-generation sequencing for routine molecular typing and pathogen characterization remains a substantial challenge due to the required bioinformatics resource and/or expertise. Therefore, the bioinformatics data analysis is still a clear bottleneck that restricts accessibility and wide adoption of NGS technologies in research and surveillance of the Mycobacterium tuberculosis. So we need tools and programs to help us for this analysis. Numerous software packages and workflows have been developed to facilitate the analysis of the sequence data, as you can see in this slide. Different steps in the analysis of next-generation sequencing data are shown in orange, for example, the assembly, the genomic characterization, comparative genomics, and phylogeny. Moreover, analysis tools are grouped by the analysis steps that they perform and are separated by user interface in shades of blue. For example, some of them do the complete analysis, some of them are web-based, and some of them are command line. However, before starting the analysis, the first step is that we should aware of the content of the sequence data from a microbial sample. As you can see here, we may have some differences between the sequence data we gain from the sequencing machine and the expected genome of bacteria that we're interested in. There can be some bias. For example, we may have a portion of contamination, or we may have some unsequenced region. So let's start with the first part of this presentation, web tools and pipelines for fast identification of bacterial strains from raw sequencing reads that can show us if we have contamination in our sequence reads or what we really sequenced. For example, if we aim to sequence Mycobacterium tuberculosis, using these web tools, we can see if we have a pure Mycobacterium tuberculosis sequence or we have a mix of Mycobacterium tuberculosis with other bacteria, or even we don't have any Mycobacterium tuberculosis in our sequence read. So the first tool is Strain Seeker, which is a program for quick classification of bacterial isolates into clonal groups or clades directly from raw whole genome sequence reads. Strain Seeker uses a guide tree to approximate phylogenetic relationship between reference bacterial genomes down to the strain level, not being tied to existing taxonomic system such as the NCBI taxonomy. So you can use the Strain Seeker web tool, which is designed for analyzing small data set on lines. The database that the web tool uses consists of more than 4,000 bacterial strains obtained from the NCBI RefSeq repository. You can also build customized database or analyze very large dataset. So if you wish to have access to more options, you should build customized databases or analyze very large dataset. Please consider using the downloadable version of the Strain Seeker and not just the web tools. But if you are doing the analysis for a couple of FASTQ files and you are not familiar with the command line environment, you can easily go to this website and you can upload your FASTQ files, and you can insert your email address so you will be notified when your analysis will be finished. Here is an example of a primary sputum culture of a patient that underwent sequencing, and as you can see, based on the analysis by strain seeker, a mixed bacterial sequence present in this FASTQ file. And if you look carefully to the names of the bacteria, you can see that various species were detected and most of them are belong to the normal oropharyngeal flora. So there is no MTBC here. Instead, we have different bacterial species belongs to the normal flora of the oropharyngeal. So here we have another example of a FASTQ file that contains not only Mycobacterium tuberculosis, but also non-tuberculosis mycobacteria. Based on the identification done by strain seeker, you can see that we have two different species. One of them is an MTM, that is Mycobacterium avium complex, or a member of Mycobacterium avium complex in this FASTQ file, and we have about 26% of the Mycobacterium tuberculosis. So in this FASTQ file, we have a mixture of NTM and MTB that strain seeker has the ability to distinguish between these two species for us. And here finally, we can see the result of a strain seeker for a pure MTBC sequence. As you can see in this FASTQ file obtained from the Illumina sequencing machine, we have only, or better to say 100%, Mycobacterium tuberculosis DNA that have been sequenced. And finally, the last example of a strain seeker analysis is a mix of two Mycobacterium tuberculosis lineages detected by this pipeline. You may ask how a strain seeker can detect two MTBC lineage. As I mentioned previously, a strain seeker uses a guide tree to approximate phylogenetic relationship between reference bacterial genomes down to the strain level. So possibly here we have two Mycobacterium tuberculosis that they are far from each other in the phylogenetic tree made by the strain seeker, and the strain seeker detected them as two distinct strains. Consider that, for example, they can be one of them from the lineage 4 that you can see here is similar to H37 RV, so we can guess that it is a lineage 4, and the other one can be another lineage a bit far from the lineage 4. As an example, one Mycobacterium africanum or a Mycobacterium tuberculosis of lineage 1 or lineage 5. So it can be detected by a strain seeker as two distinct Mycobacterium tuberculosis strains. So the next tool for the identification of the bacterial species directly from the sequence read is Kraken-2. Kraken-2 is an ultra-fast metagenomic sequence classification using exact alignment. Kraken is a system for assigning taxonomic labels to short DNA sequence usually obtained through metagenomic studies. So you can use it also for the FASTQ file from the culture positive, for example, Mycobacterium tuberculosis, or from a primary positive midget tube of a patient. The previous attempts by other bioinformatics software to accomplish the task of assigning the taxonomic labels to the DNA sequences have often used sequence alignment or machine learning techniques that were quite slow, leading to development of less sensitive but much faster abundance estimation programs. Kraken aims to achieve high sensitivity and high speed by utilizing exact alignment of k-mers and a novel classification algorithm. In its fastest mode of operation, for a simulated metagenomes of 100 base pair reads, Kraken processes over 4 million reads per minute on a single core, over 900 times faster than MegaBlast and over 10 times faster than the abundance estimation program MetaFlan. Kraken accuracy is comparable with MegaBlast with a slightly lower sensitivity and very high precision. The Kraken is written in C++ and Perl and is designed for use in the Linux operating system and also Mac OS. And please note that the Kraken 2 is the newest version of Kraken, and Kraken 1 will continue to be available via the Kraken 1 GitHub page, but is not longer being supported. So if you want to install it, please pay attention that you have to install the Kraken 2. And one limitation of the Kraken 2 that I would like to mention is that the Kraken 2 is only available in command line interface. So for you microbiologists or laboratory lab people who may not be familiar with command line, it can be difficult to install and use it because you need to install the Kraken and then you need to install the database of the Kraken so you can do the analysis of your samples. But don't worry because there are some good people that they have made it easy for you. I will show you in the next slide. Then you can use, for example, Patrick in order to access directly to the Kraken without any problem of installation or the speed of analysis and, for example, downloading and upgrading the database. Just what you have to do is to upload your FASTQ files, and after a couple of minutes you will see the results and you can visualize your results in a very details of the output. So in the next slides I will show you how to do it. So to overcome this issue, I'm going to introduce you the BVBRC, or the Bacterial and Viral Bioinformatics Resource Center. That is an information system designed to support research on bacterial and viral infectious diseases. BVBRC combines two long-running bioinformatics resource centers, the Patrick, or the bacterial system, and the IRD VIPR, the viral system. It's freely available and you can upload your private data in a workspace, analyze it using high throughput services, and compare it with the other public databases using visual analytical tools. So Patrick is available in this website, patrickbrc.org, and after you make your profile, you can use the Patrick freely and without any limitation. So you should upload your data, and as you can see, a wide variety of file formats are supported by Patrick. We are going to use the Kraken, so we need to upload our FASTQ files here, and when you select your FASTQ file, you should assign the format of the file, that is reads for the FASTQ files, and you can so in order to use the Kraken, you can go to the toolbar and select the services, and from the services you should select the taxonomic classification. The taxonomic classification service computes taxonomic classification for the sequence read data using the Kraken tool. So you can see that you don't need to use the command line so to be able to use the Kraken. You can directly use the Kraken from the Patrick without any problems and without any costs. So when you click on the taxonomy classification, you will be directed to this page that is for the Kraken analysis inside the Patrick tool. The taxonomy classification service accepts reads or counting, it means the assembled genome, from a sequencing machine, and to assign the reads to taxonomic beans, providing an initial profile of the possible constituent organism present in the sample. Input file depending on the option chosen above. Here you can see read file or assemble counting. So the input file section will request sequence reads or assemble countings respectively. So here you can see the output of a Kraken analysis of a FASTQ files. And as you can see here, we have 99% of the sequence reads which belongs to the Mycobacterium tuberculosis. And we have 1% of the noise that can be some contamination present in the culture, or some contaminations happened during the DNA extraction or library preparation. But we can consider this FASTQ file as a high quality FASTQ files. In contrary, here you can see a result of the primary midget culture that, if you compare to the previous slides, it's totally different. We have a lot of bacterial species detected by Kraken that, as you remember the analysis by strain seeker, these bacteria are, most of them belongs to the oropharyngeal normal flora that is normally present in the sputum sample collected from the patients. And here you can see we have even 4% of the human DNA and 38% without any hits. It means that or the DNA was degraded during the sequencing, during the library preparation and sequencing, so no bacterial or no organism DNA is similar to this, or there are belong to some organisms or microorganisms that have not sequenced yet. So we don't have any information from this microorganism or organism in RefSeq. So the Kraken cannot assign any classification for this read. So in general, we consider this sequencing file of a bad quality and without detection of Mycobacterium tuberculosis, even in 1% in this FASTQ file. And here you can see the last example provided for you. You can see that Kraken detected a combination of Mycobacterium tuberculosis complex and NTM non-tuberculosis mycobacteria, that is Mycobacterium avium complex, in this FASTQ files. So we have about 2/3 of Mycobacterium tuberculosis in this FASTQ file, and 30% of the sequence reads belong to the Mycobacterium avium complex. So you have seen that with both strain seeker and Kraken, we are able to see what we have in the sequences that we obtained from the sequencing machine. So in the next part of this presentation, we will review some web tools and software designed for the analysis of the Mycobacterium tuberculosis sequence data. By using these tools, you will have information of the drug resistance pattern and the lineage information of the sample that you have sequenced. To date, 3 web servers, CastTV, FireS, and TV Profiler, and 2 software solutions, CoverQ and Microbe, have been developed to enable non-specialists to infer drug resistance from whole genome sequencing data. Moreover, all tools provide epidemiological typing results, which might be used for contact tracing or surveillance, and can affect patient treatment directly if a member of the MTBC is identified that is naturally resistant to one or more TB drugs. So let's start with one of the user-friendly tools for drug-resistant TB diagnosis by sequencing data, which called Microbe. But how does Microbe work? Microbe analyzes the whole genome sequence of a bacterial sample all within a couple of minutes and predict which drugs the infection is resistant to. It has been heavily validated on thousands of samples, is free, no expertise is needed to run or interpret it, and it works offline on a standard desktop or laptop. So you can easily go to this website and you can install the Microbe in your computer. And after installing the software in your PC, for the analysis, you won't need internet connection. Microbe supports both traditional sequencing machine or Illumina, which tends to need a large hospital or lab and a lot of infrastructure, and also the new handheld sequencers or long-read sequencing by Oxford Nanopore, which are very portable and moving toward production readiness. So, as I mentioned, you can install the Microbe, and after installing, you can open the software and you can see here, you should upload your file. Microbe can analyze 1 sample per time. So you should upload your file, and after uploading, the analysis will start by constructing the genome, as you can see here. And after the analysis is finished, you will have the information on the drug resistance. You can see, for example, in this sample, we have a sample resistant to rifampicin, ethambutol, and pyrazinamide, but susceptible to isoniazid. And for the second line drugs, you can see that we have resistance to fluoroquinolones, but susceptible to streptomycin and other antibiotics. If you are looking for the evidence of these resistant patterns, you can see, for example, for the fluoroquinolones, for example, ciprofloxacin, moxifloxacin, and ofloxacin, we have a D94G in GIR-A. So this is a high confidence mutation that have been found in this sample. And you can see the depths, the information on depths of the allele. You can see here the depths of 40 on the resistant alleles. It means that all the sequence reads in these samples support this mutation, and they had this nucleotide change that leads to this amino acid change in codon 94 of the GA. You can see also for the rifampicin that we have the L452P mutation, another high confidence mutation in RPOB. And in pyrazinamide, we have a T insertion in the gene of PNCA. But as you can see here, in 27 alleles or 27 reads, we have this allele. In 9 alleles, we have the wild type. It means that for the pyrazinamide, we have a heteroresistance or two population of the MTBC in this sample. The majority are resistant to pyrazinamide by having this insertion in the PNCA, and the minority, which are wild type in PNCA, are susceptible to this drug. Moreover, if you click on the species, you will have information on the lineage and the species. That here you can see that the samples that were sequenced were a Mycobacterium tuberculosis belongs to the lineage 3. So the next tool that we will review today called TB Profiler. TB Profiler is a pipeline which allows users to analyze Mycobacterium tuberculosis complex whole genome sequence data to predict drug resistance pattern and the lineage. The TB Profiler search for a small variant and big deletions associated with drug resistance. It will also report the lineage. TB Profiler by default uses Tree-O-Matic to trim the reads. It used for the mapping of the reads to the reference genome, BWA for Illumina short reads, or Minimap2 for nanopore line reads. And it used the GATK version 4 to call the variant. And I'm sure that you have followed the previous webinars how to analyze the sequence data, so you are familiar already with BWA and GATK and other tools that are used for the downstream analysis of the sequence data. So in TB Profiler, you can go to this website, tbdr.ishtm.ac.uk, and you can upload your next generation sequencing data in FASTQ format. You can upload one or two, for example, forward and reverse FASTQ file. And when you upload your data, the run will be assigned with a unique ID. And you have to take a note of this unique ID because you will need to find your results later. And batch upload of the samples is also available. If you want to analyze, for example, a batch of 10 or 20 samples, you can do it simultaneously in TB Profiler. So, a question is that which mutations are being used to predict the resistance by TB Profiler? The resistance is predicted using a curated database that has been tested using over 17,000 samples with genotyping and phenotypic data. The next question is that, can I run TB Profiler on my own computer instead of using as a web tool? The answer is yes. If you have access to a Linux or OS X operating system, then you can download the command line version of the TB Profiler. And for the more information on this installation and how to install, how to run, and how to set up the TB Profiler in command line environment, you can visit the GitHub repository of the TB profiler. You go to the GitHub and you search for the TB profiler, and you will have all information on installation, setting up, and the usage of TB profiler for those who want to use it in command line. But for those who want to use as a web tool, you can easily go to this address and you can upload your FASTQ file, as we can see in the next slide, and we will see the interpreted results for the drug resistance and the lineage. So as you can see, you can upload your next generation sequencing data in FASTQ format. Here, for example, I'm going to analyze a FASTQ file from a patient, from a pre-XDR patient under treatment with bedaquiline containing regimen. And these samples were obtained from a positive culture after 9 months of treatment with bedaquiline containing regimen. So you may select also the technology used to generate the data. If is it Illumina or Oxford Nanopore. The samples will be processed with the first in first out policy, so please be patient as there may be runs waiting to be processed before yours. And please note that at the moment, the TB profiler can only accommodate uploads of under 1 gigabyte. So if you have files which are larger or you require many isolates to be processed and have access to a Linux or OS X operating system, then it might be worthwhile to run the command line version of the TB profiler. As I mentioned in the previous slide, you can have more information on these on GitHub repository of the TB profiler. So here you can see that unique ID that I mentioned, that is for your analysis run. So you have to conserve this unique code, and whenever you want, you can back to the website, and by inserting this ID, you can see your results. However, you can also wait until the analysis is finished. You can see here, for example, in this step, the mapping is going to done, and you can see here your file are being processed, and please come back later. So you can close also your browser and you come back later. The analysis will not be interrupted when you close your browser. So here you can see information of the analysis after finishing. You can have the information of the number of the reads. Here in this case, for example, we have more than 2.6 millions of the reads, and you can have information on the percentage of the reads mapped on reference genome. In this case, we have almost 99%. That this can be also one indication for you to see if you have pure sequence of MTBC or not. For example, if you have 50% of the mapped reads, so you will understand that something is going wrong. So you don't have 100% Mycobacterium tuberculosis complex in your sequencing data. So this is also one important indication showing that how pure your sequence is. And you can see that this strain belongs to the lineage 3, and the drug resistance pattern is pre-XDR. And here you can download in different formats the analysis done by TB profiler. So here you can see the evidence of why this sample diagnosed as pre-XDR by TB profiler. You can see that we have a well-known S315T in CAT-G, S450L in RPOB, and M306 isoleucine in ethambutol. And for PNCA, we have an insertion. And for example, for streptomycin, we have also a high confidence mutation in RPSL. And also for fluoroquinolones, we have A90V in GA. So this sample, based on these mutations, high confidence mutation is a pre-XDR. And next, you can see the information of the lineage. You can see this is a lineage tree, East African Indian. The main oligotype is Cas, the RD, for example, RD715, and the frequency is 1. It means that we have only one strain and not a mix of 2 TB in our sample. So here you can see the drug-resistant associated mutation. These tables report mutation found in candidate-resistant genes which have been associated with drug resistance. As I mentioned previously, these are the high-confidence mutations in the genes associated with resistance to isoniazid, pyrazinamide, ethambutol, fluoroquinolones, rifampicin, and streptomycin. Next, we have non-associated mutations, that this table reports mutation found in candidate-resistant genes which have not been associated with drug resistance. As you may be familiar with some of them, for example, this mutation in CAT-G is a polymorphism and is a phylogenetic related SNPs, so has not anything to do with the resistance. However, if you remember, I have told you that this sample obtained from a patient, from a pre-XDR patient under treatment with bedaquiline containing regimen and 9 months. So you can see here we have in MMPR5 or RV0678 and gained a stop codon. And the MIC of bedaquiline for this sample in midget is 2 mg per liter, while the bedaquiline MIC for the baseline isolate was 0.5 mg per liter. So we have seen a 4-fold increase in MIC in these samples, and we have found an stop codon in the gene related to resistance to bedaquiline. So we can interpret this mutation as resistant to bedaquiline and this sample resistant to bedaquiline. So it's not a pre-XDR anymore, and based on the new definition is a XDR because it's resistance to a group A drug that is bedaquiline.
So the last tool I'm going to present to you today is called FIRES. That is a simple and reliable viewer of mycobacterial DNA reads. FIRES is a web server for lineage identification and resistance prediction based on raw whole genome sequencing data. FIRES is available from its website, that is www.fires.org. The mutation catalog of FIRES is based on both a literature review and experimental data. The catalog comprises of more than 300 variants to predict resistance to 12 antibiotics and 239 single nucleotide polymorphism in 135 genes for phylogenetic typing. So here you can see the workflow of the FIRES. Any pair-end or single-end data stemming from Illumina or Ion Torrent next-generation sequencing of Mycobacterium tuberculosis can be analyzed by FIRES. This web tool combines well-established methods from FastQC, BWA, QualiMap, SAMtools, and other tools with the treasure trove or experimentally validated cases acquired at the Institute of Research Center, Borstel. The reports on strain lineage, and antibiotic resistance will be provided at the end of the workflow. As you can see here, we have sequential steps at FIRES accessible to the users of one specific sessions that you can see in red. Input to the list of known mutation is shown in green. So from the literature, from experiments, and from other sources of data, the mutation were gathered into the dictionary of mutation in FIRES. And the list of consists of common resistance and phylogenetic SNPs extracted from the literature used for the drug resistance prediction and for lineage assignment in FIRES. So here you can see the upload page of the FIRES where you can upload your files you wish to analyze. You can upload several files simultaneously and the analysis will be done by the FIRES. Please note that you need a good speed of internet in order to upload your file in a fastly manner. And also, the same as TB Profiler, here also a key or an ID will be assigned to your run. So you have keep this ID as you can see here. So after the analysis or after a couple of days or week, if you want to come back to your results by inserting this session key into the web page of the FIRES, you will be directed to the results.
So once the upload process is finished, the analysis will start and you will be directed to this page that you can see the list of the samples uploaded and they are going to be analyzed in the FIRES. After the progress finished, you can see different steps of the analysis. The first one is the FastQC, that you can see some clues on the quality of your FASTQ file, and this one is for the mapping, and this one is for variant calling. The other step is the lineage identification, and the last step you can see the drug resistance prediction of the samples. So for each sample, 5 steps will be done: the FastQC, the mapping, variant calling, lineage identification, and drug resistance prediction. As an example, here you can see the FastQC analysis of one of the samples. You will have the information of the total number of the reads, in this case 1,300,000 reads, and about the sequence lengths that you can see 35 to 151, as we were using the short read, so Illumina, so you can see that the read length and the GC content. GC content also is very important indicator of our sequencing. As you know, Mycobacterium tuberculosis is among the high GC content bacterial species. So consider that after the QC analysis, you see a GC content of, for example, 40%, and you can be suspicious of the bacteria that you have sequenced, because MTBC should have a GC content above 65%. So when you see 40%, it means that you don't have MTBC in your sequence data. And the next step is mapping. You can see here we have the reference genome. Here we have the reference genome that is H37RV, and here are the sequence reads that you can see like pieces of a jigsaw puzzles. It is mapped on the reference genome to reconstruct the genome. So also the mapping step, we can get some important information. For example, here in this position, as you can see, and as you remember from the previous webinars of the introduction to the analysis steps, you can see that here we have the reads and here we have the reference genome. What you can see here is that in this position, that is the position 2155168, we have a nucleotide change. As you can see in reference genome, we have nucleotide cytosine, but in our isolate or strain, sequence strain, we have a G. This position is the coordinates of the codon 315 in cat-G, and this nucleotide change results to the amino acid change because it's in the coding region of the cat-G. So you can see how the variant calling step interpret these mismatch in the BAM file and call it as a variant, and you can see in the variant call file. And the annotation for this mutation is cat-G in codon 315, the amino acid serine changed to teroid. As you know better than me, this is one of the most famous mutation and a high confidence mutation in cat-G that is associated with resistance to isoniazid. Then let's take a look at the drug resistance prediction by the virus. If you can see in these samples, a total number of 5 high confidence mutation. Why high confidence? Because you see that here we have a column of high confidence SNPs, and the answer for all of them are yes. So it means that all of these mutations are associated with resistance. So in our samples, we have found 5 mutations related to drug resistance. You can see here that we have one mutation in coding region are RV0667 or RPOB in the codon 415. We have this nucleotide change, one cytosine changed for a thymine, and this is results to the amino acid change S450L that is well known as causing resistance to rifampicin. Moreover, we have another mutation in -15 of the INHA FABG1 intergenic region that is associated with resistance to isoniazid. So until now, we have found MDR-TB having resistance to rifampicin and isoniazid. But as you can see here, we have also other mutations. One of them is in cat-G and 2 in EMBB. The difference of this mutation with these 2 mutations above is that the frequency of these mutations, as you can see here, is 42% compared to the RPOB mutation, which is 97%. So 42% of the population of these bacteria that has been sequenced has this mutation. So we have a heterozygous variant, or we have a heteroresistance variant for this gene in our sample. So it means that 42% have S315 in cat G, but 58% of the population are wild type in this specific sample. The same for ethambutol. You can see that we have found two minor variants, one of them in EMBB with 33% and the other one is in cat G. So these are also very important to attention and to make it to the consideration when we are interpreting the results. Just finding the variance is not important. More important thing is to look at the variance, to gather all the knowledge we have about the variance, and to make the right and the 100% correct interpretation of the variance that can save the life of the patients. And the last step of FIRES is the lineage identification or genotyping step. As you can see, the samples that we were analyzed is a Dedicas lineage. And as you know, Dedicas is belong to the lineage 3. So by FIRES, by TB profiler, and by microbe, we can have information on drug resistance and genotyping of our samples. These tools are free to use, friendly user, and can be the first step for you as microbiologists, as biologists, and laboratory personnel to start step by step doing the analysis of your sequence data.